Hi there, welcome to CM Explain Science Hub. In this video, we are going deep into RNA transcription, the fundamental biological process where cells convert genetic information from DNA into RNA. Whether you're preparing for biology exams or building your understanding of gene expression, this video covers everything. Don't forget to subscribe and like the video. What is RNA transcription? So RNA transcription is the biological enzymatic process where a segment of DNA is used as a template to make a complementary RNA molecule. To be specific, that is the messenger RNA. This process is catalyzed by the enzyme RNA polymerase. Now, we talked of copying from DNA to RNA. Now, the segment of the DNA in which transcription takes place, it is called a gene. And in this gene, once the double helix structure of a gene is separated, we'll learn further in under initiation and other processes. Once they are separated there, the two strands, they're named, we have the coding or the sense strand, and the other one is the template and the antisense strand. Let's now talk about the parts and functions of the part of the enzyme RNA polymerase. So it is made up of 1 sigma subunit, 2 alpha subunits, 1 beta subunit, and 1 beta prime subunit. The functions of the sigma subunit is that it recognizes the promoter and guides the RNA polymerase to the correct starting site so that transcription might start to dig deeper when we talk about initiation. For the 2 alpha subunits, their function is they interact with regulatory proteins as the DNA is being transcribed. The beta subunit is used to catalyze phosphodiester bonds joining the RNA nucleotides together. Under nucleotides, we know to say a nucleotide is joined to another nucleotide with a bond, the phosphodiester bond. So the one that is responsible there is the beta subunit. For the last part or the last subunit, the beta prime, it binds to the DNA. So this avoids the enzyme going out of or away. So it holds on to the DNA as it is moving and doing its role as the enzyme for transcription. Now, let's talk about the stages of transcription. The first stage in transcription is initiation. Under initiation, we have the recognition of the promoter, which is done by the sigma subunit. So it recognizes 2 sequences, and the sequence that it recognizes is called the promoter. Under this promoter, we have 2 sequences. We have the negative 35 and negative 10, which is also called the data box. Now, once it recognizes the promoter region, it attaches itself to the negative 35 sequence. And once attached there, now the whole enzyme, the core enzyme, comes looking for the subunit, and it attaches the sigma subunit back to the enzyme, and hence the enzyme now knows where it needs to start the recognition from. The negative 10 box, it contains the adenine and thymine nitrogenous bases, which have 2 hydrogen bonds between them. And those 2, in comparison to the other G and C, they are very weak. And so Using that same region which has which only has A and T, it is considered to be very weak, and so that is where now the separation of the two strands, the sense and the antisense strands, starts from. Once the coenzyme now combines with the sigma subunit, it forms a holoenzyme. Now this holoenzyme in the promoter region creates what we call as the closed complex. This closed complex, and when we have the tata box there which opens, it now creates the open complex. We look at the term consensus sequence. So a consensus sequence is a sequence representing the most frequent nucleotides found in a certain region. For example, for the -35, it is called the TTGACA. So those are the nucleotides that are most frequent in that region. And for the data box, it is only A and T. The TSS, or the transcription start site. So this is a point. Usually when we have those promoter diagrams, it's written +1. So that is the point, or the nucleotide, where the transcription starts from. As the name suggests, transcription start site. The direction of the transcription is the same as the one in replication, from the 5' to the 3'. So the 5', by adding the enzyme, so 3' of the first enzyme, and hence once we have that long one, we're moving from the 5 to the 3'. Let's not confuse ourselves on the 5' and 3' directions. We should also note that this process that we're talking about is basically in prokaryotes, because they are simpler than eukaryotes. Though the process is just the same, such that in eukaryotes we have different things that, for example, the TSS, it is different in eukaryotes than in prokaryotes. Now, let's talk about the Tata box. So this Tata box, in other words, they call it the Pribnow box. It having that 80 bonds being weak, and once they're separating, it is classified as melting. So the melting of the Pribnow box. Now we talk about the second stage of transcription, which is elongation. Elongation, this is the synthesis of the full RNA transcript, which is done by RNA polymerase coenzyme, using the template strand in the direction of 5' to 3'. The RNA nucleotides are added to the 3' end of the transcript. Another feature that we need to take note of is the RNA-DNA hybrid region. So this is the region having RNA and DNA before the RNA detaches itself from the template strand. It has about 8 to 9 base pairs. The transcription bubble. This is a temporal region where transcription occurs. It moves in the same direction, 5' to 3' as earlier stated. It does its transcription. This is a site where transcription occurs, and how does it do its work? So the unzipping of the DNA happens there in this same transcription bubble. Once it unzips, we attach the new RNA nucleotides to the antisense there to make the new mRNA there. And also, that is the same region where we have the 8 to 9 RNA-DNA hybrid base pairs. Once the RNA has been made and it is moving out, the two remaining DNA strands, the sense strand and the antisense strand, wind up again, forming the double helix DNA structure that we know. And also the length of this transcription bubble, it has about Others say 12 to 14, other books say 14 to 17. So in that range, let's just say 12 to 17 base pairs in the same transcription bubble. The last process of transcription is the termination process, as the name suggests, terminating meaning the end. So the RNA molecule is complete and it separates from the template and the core enzyme. And the enzyme now falls off and goes to look for the sigma bond so that we have another transcription at another point in a gene. Termination happens in the region called the terminator region, which is a region with DNA sequence that indicates the end point of transcription. And we should take note to say the terminator are also copied into the RNA transcript. So it is there in the DNA, and also in the transcript we also find a terminator there. Now, termination being the end, it happens in two ways. So we have the intrinsic termination, which is in other ways called the Rho-independent, and also the Rho-dependent. Let's talk about intrinsic or the Rho-independent termination. So this is the type of termination that does not need an external factor, but stops by itself. The DNA is a special sequence. The DNA has a special sequence of G and C base pairs, followed by several A or adenine base pairs. The G and C are transcribed onto the RNA, and so are the A, which in terms of the RNA will be the U or the uracil. Now, the G and C, we know to say G and C forms three hydrogen bonds, which are strong as compared to the A and U. They create a loop. So they bond to each other. This same sequence, the C and the G, they come together, create that hairpin type of structure there. And that itself is called the stem loop. Now, these the G and C coming together, creating that stem loop, it weakens those A and U bonds which proceed from the G and C sequences. And since this A and U are weak, they are disturbed by this previous G and C sequence. Yeah, and another point we need to take note of is the A and U, the A base pairs usually run from 5 to 10 nucleotides, or nitrogenous bases there. And also this same region, where we have the G and the C, this is where now the pausing of the RNA in the other type happens. And the other type of termination is the Rho-dependent. So this Rho-dependent, as the name suggests, it is dependent on Rho. So it requires assistance of a protein called the Rho. And this Rho is a type of a helicase protein. And when we remember in the video replication, we talked about the function of the helicase as being the unzipping there. So the Rho, it unzips the RNA-DNA polymerase. Now, we have another structure which is known as the RUT site. So the Rho utilization site. So this is like the starting site. It is a sequence of on the RNA transcript where the Rho attaches itself and starts to move. So let's have this example to say we have a person A and person B. So person A starts from the starting point in a race. They are racing. Then person B starts to follow or to chase that person B. And the starting point of this person B being, this person B is the Rho protein. It starts from, let's say, halfway. Yeah. So that point where let's say it's 100 meters, the 50 meters there, that is called the RUT site. So in biological terms, can be the site where the Rho starts the chase from. So it starts chasing the the RNA polymerase. And once the RNA polymerase reaches the finishing line, which in this case is the terminating region, where we talked about the GC concentration, like we have a lot of those, and the 5 to 10 A base pairs there. So once it reaches there, the polymerase there is going to slow down. And now the person behind, or the Rho protein, will now catch up, seeing that the enzyme and the Rho protein are moving at the same speed. Once the other one pauses or slows down, the Rho protein catches up and it unzips the DNA RNA hybrid, which we say that it is a helicase, and hence the unzipping there. Lastly but not the least, we'll talk about palindromic sequence. So the palindromic sequence is a DNA or RNA sequence that reads the same forwards and backwards on complementary strands in the 5 to 3 prime direction. Let's take, for instance, we have a strand which we say it is written as GAATTC. The complementary base pair is going to have CTTAAG. But if we read both of them in the same direction as the 5 prime to 3 prime, the first one is going to read GAATC. The second one is also going to read GAATTC. So they are the same but different. And when they go in the same direction, they are the same. When they are in the way they're supposed to be, like in DNA, they are different, because one is running from 5 prime to 3, the other one is 3 to 5. So that is basically that on palindromic sequencing. And that's a wrap on RNA transcription. From the promoter to the terminator, we've seen how cells turn DNA into RNA using powerful machinery of RNA polymerase, the sigma subunit, and all those clever regulatory sequences. Whether it is the Pribnow box, the transcription bubble, or the stem loop terminators, every step is a finely tuned process essential for gene expression. If you found this helpful, don't forget to like, subscribe, and hit the notification bell so you never miss out on our clear, simplified breakdowns of biology and chemistry topics. Got any questions? Drop them in the comments below. Until next time, keep exploring, stay curious, and remember, science makes the invisible understandable.